Over the past few years, a growing body of research has explored the potential of rapamycin as a therapeutic agent capable of extending lifespan and slowing down the biological process of aging. Initially discovered in the 1960s in a soil sample from Easter Island, rapamycin was first recognized for its antifungal properties. It wasn't long before scientists realized that it also had potent immunosuppressive effects, leading to its use in organ transplantation. Despite its potential to save lives by preventing organ rejection, rapamycin also came with significant risks, particularly for individuals whose immune systems were already compromised. By the late 1990s, rapamycin was approved by the FDA for use in organ transplant patients, where it was primarily employed to suppress the immune system and reduce the risk of organ rejection. However, it wasn't until 2009 that researchers began to make a connection between rapamycin's biological mechanisms and aging. In that year, a landmark study on older mice showed that rapamycin could increase lifespan by 14% in females and 9% in males. Further studies suggested that combining rapamycin with other drugs, such as metformin, could even yield greater longevity benefits, particularly for females who showed a 28% increase in lifespan and males who experienced a 34% increase. As these studies in animals gained attention, hopes rose that rapamycin might be a viable intervention for humans seeking to delay aging and prevent age-related diseases. Rapamycin's potential in this regard is linked to its ability to inhibit a critical pathway in the body known as mTORC1, mechanistic target of rapamycin complex 1. This pathway regulates growth and cell proliferation, and overactivation of mTORC1 has been implicated in various age-related diseases, including cancer, neurodegeneration, and cardiovascular conditions. Naturally, interventions that inhibit mTORC1, such as caloric restriction and exercise, have been associated with longevity, and rapamycin's ability to directly target this pathway made it an intriguing candidate for anti-aging therapies. However, despite its promise, rapamycin is far from a panacea. The drug also inhibits mTORC2, mechanistic target of rapamycin complex 2, a related pathway involved in cellular processes that help maintain metabolic balance. Inhibition of mTORC2 has been associated with a range of side effects, including metabolic disruptions, high cholesterol, and insulin resistance. Furthermore, rapamycin's immunosuppressive properties can have serious consequences, as it weakens the immune system and reduces the activity of NK, natural killer, cells, those responsible for fighting cancer. The potential for increased cancer risk is a critical concern when considering long-term use of rapamycin as an anti-aging intervention. Despite these known risks, recent studies have shown that lower doses or intermittent dosing of rapamycin may mitigate some of the side effects while still providing potential benefits for longevity. This balance between achieving the desired anti-aging effects and minimizing the associated risks has become a focal point for ongoing research. In the context of human trials, early results have been mixed. While some studies show positive effects on aging markers and longevity, the side effects have proven to be a significant challenge. For instance, some individuals have reported issues such as impaired wound healing, increased cholesterol levels, and elevated blood glucose, all of which are concerning when considering the long-term health implications of rapamycin use. These adverse effects are particularly troubling because they may outweigh the potential benefits, especially if the drug accelerates aging in certain populations or under certain dosing regimens. In late October, a preprint study surfaced, which provided a sobering analysis of rapamycin's impact on human aging. The study evaluated several longevity interventions, including rapamycin, and found that, contrary to expectations, the drug appeared to accelerate the biological speed of aging in humans, as measured by 16 different epigenetic clocks. This finding was a significant blow to the hypothesis that rapamycin could be the breakthrough drug for extending human lifespan. The study's results suggest that rapamycin may have unintended consequences, exacerbating aging rather than mitigating it, and raising critical questions about its suitability for long-term use as an anti-aging therapy. Although the study's findings have shaken the enthusiasm surrounding rapamycin, it is important to remain cautious and recognize the complexity of scientific research. There is always the possibility that the study was flawed, or that rapamycin still holds promise if administered under specific conditions or combined with other treatments. This highlights the ongoing uncertainty in the field of longevity research, where interventions often yield unexpected results and require careful, long-term observation. For those who are considering the potential of rapamycin as an anti-aging intervention, it is essential to be aware of the risks. The drug's impact on the immune system, metabolism, and cancer risk cannot be ignored. Furthermore, the potential for accelerated aging, as suggested by recent research, serves as a warning that even drugs with seemingly promising results can have unforeseen consequences. As with all experimental therapies, further studies are needed to clarify the long-term effects of rapamycin and determine whether it can safely be used as part of an anti-aging regimen. The complexity of aging and the multitude of factors that influence longevity means that there is no one-size-fits-all solution. While rapamycin may eventually prove to be a valuable tool in the fight against aging, its current trajectory underscores the importance of cautious optimism and scientific rigor. As the field of longevity research continues to evolve, it is essential to carefully consider both the potential benefits and the risks, and to continually assess new evidence as it emerges. The quest for longer, healthier lives will no doubt benefit from this ongoing process of discovery, but it will also require a willingness to embrace both success and failure along the way.